So we're going to go ahead and revisit the cell membrane, okay, and recall the plasma membrane, the proteins, and everything else that's embedded in it, and what allows for that transport of material across the membrane itself. So remember, it's made up of two layers. It is a phospholipid bilayer, okay? It has hydrophilic heads, which actually do love water, and they're not scared of water, so hydrophilic phosphate heads. And then they have the hydrophobic tails, which actually interact and face one another because they do not want to touch water at all. Okay? It is called a fluid mosaic model because mosaic meaning it's a mixture of all different kinds of components such as phospholipids, steroids, proteins, carbohydrates. Okay, and it is called fluid, okay, mosaic because it is actually very it does have a lot of fluidity, and it is very mobile. It can move around. Remember, those phospholipids can jump around. They can flip on each other. They can also exchange sides with one another. So it's called the fluid mosaic model. So some of the plasma membranes have all different kinds of integral membrane proteins, which actually attach to the bilayer itself. Okay, it goes all the way through the plasma membrane, remember? Okay, so basically right here, these are all examples of integral membrane proteins, like this one right here and this one right here. Adhesion proteins fasten cells together or to their external proteins themselves. This one right here, you can see it, it's very, very dim down here, but you can actually see it's going all the way through from one side to the other side of the actual plasma membrane itself. We also have receptor proteins right here, which is, as it explains here, it triggers a change in the cellular activity in response to a stimulus such as binding to a particular substance. So something's going to bind over here to this receptor, then that's going to actually signal and transfer that communication into the actual cell for another reaction to occur. But it is going throughout the entire membrane. We have transport proteins right here, which are actually kind of like a channel going through the plasma membrane, allowing things into and out of the cell as needed. Okay. So, and we actually just finished talking about enzymes last lecture. This is basically enzymes. Remember, they help with catalysis, or they help reactions occur a lot quicker. So diffusion, okay? Diffusion is the actual movement of solute molecules down their concentration gradient. So they're going to go from a high solute concentration area to a low solute concentration area. The rate of the diffusion depends on these 5 factors: on the size of the molecule, because a lot smaller molecules are going to move a lot quicker than larger ones. Temperature, okay, because if it's colder, those molecules are going to move a lot slower, but if it's a higher temperature, they're going to move a lot quicker. The concentration gradient, so the greater the amount of solute you have in a solution, the obviously quicker that is going to actually diffuse across the entire new solution. And the charges affect the rate and direction as well in diffusion. Pressure does often occur, diffusion actually will occur quicker at higher pressures than in lower pressures as well, because as you can imagine, it's kind of like you're just pushing all these molecules, giving them more pressure to be pushed through to the other side of a membrane. Okay, and diffusion is known to be selectively permeable, which means that the cell membrane itself can control what substances go in and out of them. Okay, but it does allow for larger substances and other solutes to come through other than actual water itself. So diffusion, okay, remember, it's going to go from a high solute concentration to a low solute concentration. And this is a permeable membrane, which means things can go through it because of the fact that we do need larger molecules to be able to go through right here. Okay, and at the very end, it's going to basically reach equilibrium state when it's completely evened out on both sides. Okay. Diffusion across the plasma membrane will allow for hydrophobic molecules such as steroids, fats, and oils, also some gases, and small uncharged polar molecules to come across. Okay. The atoms that cannot go across with diffusion are going to be ions such as sodium and hydrogen, and this bicarbonate right here. Large uncharged polar molecules. Okay. These are the ones that are not going to be allowed to diffuse across. Osmosis though, so remember, diffusion allows for solutes to move across a permeable membrane. Diffusion is responsible for allowing water molecules to move across a semi-permeable membrane. Okay, semi-permeable means that it is very, very distinct as to what it will allow to cross or not. So it only allows usually a lot smaller molecules, which is why usually only water is permeable to a semi-permeable membrane. Okay, so right here, what you guys see, on the other one, in diffusion, we allowed for, we were looking for the solute concentration to go from a high solute to a low solute concentration. Okay, in this case though, you can see, let's just look at this beaker right here. Okay, we have a higher solute concentration on this side right here because this is going to be sugar molecules. So there's a higher sugar concentrate over here than over here because there's only one sugar molecule over here. So in diffusion, what would happen if this was a permeable membrane? These guys would come across this way to basically even out and reach equilibrium of the solution. But this is osmosis. So these sugar molecules cannot move across this semi-permeable membrane. The only thing that can move across is going to be water. So the way osmosis works is it's once again trying to reach equilibrium. But what happens is instead we're going to be moving the water molecules, which are the little blue ones down here, and it's going to move from a high water concentration, which also has low solute concentration. So this has low solute or the low sugar concentration. That means that it actually has way more water available here. So let's say this is like 1% of sugar concentration. That means there's 99% water. So water is going to move from the higher water concentration or the low solute concentration, and water is going to move to the opposite side, which is towards the low water concentration or higher solute concentration. However you want to look at it, but it's exactly the same thing, just different situations. So now water is going to go from this side, high water concentration, to over here on this side, the high solute concentration. And it's going to do that because it's trying to even out this these two solutions. So eventually they'll both be the same exact concentrations on both sides. So the water is going to be pushing over here, kind of spacing these guys out until eventually it reaches the point of equilibrium where these two concentrations, even though they have different number of sugar molecules, because one has added more water, they'll eventually be the same exact concentration on both sides. Okay. Now we're going to be looking at the different tonicities are available, whether you're talking about animal cells or plant cells. Okay? Tonicity is the relative concentration of solutes in two different fluids. So in this case, we're going to be always thinking about cells and then the actual solution that's surrounding the cells and what happens to them according to what that solute concentration is. So let's pretend right here we're looking at this one right here. This is going to be a cell. That cell has 2% sucrose inside of it. Okay, so if it has 2% sucrose inside, and over here in the solution that you're putting it in, the solution, the beaker actually has 2% sucrose as well. That would be considered you're putting the cell in an isotonic environment, basically meaning the same solute concentration in the cell as outside the cell. So this is considered an isotonic environment, so they're the same. This one, on the other hand, would be considered, let's take a look at it. If you have 10% sucrose in the beaker and 2% sucrose in the cell, that means that the solution in the beaker is hypertonic, meaning it has a higher solute concentration. So the beaker has a hypertonic solution compared to this cell. Over here, you actually have a hypotonic solution because we have 2% sucrose in the cell and 0% sucrose in the actual beaker. Okay, so what's going to happen here is you're going to say that the cell is placed in a hypo or lower solute concentration solution. Okay, so what happens when you put the different cells in each of these different tonicities? Okay, so when you put these cells in each of these different tonicities, this is what's going to happen. First of all, let's go ahead and make sure this is right here. Okay, let's go ahead and take a look at it like this. This animal cell, or red blood cell, is actually placed in this hypotonic solution. Remember, hypotonic means that the solution on the outside of the cell, on this beaker, is less than, okay, what's inside of the cell. So what's happening is osmosis wants to go from an area of low solute concentration to high solute concentration. It's trying to even it out. So water is going to move into the cell right here, but the problem is with animal cells, such as our own, it can actually eventually burst if it becomes way too engorged with water. This is called lysing a cell, and lysing, L-Y-S-E, is cell death. Okay, so that's what happens with animal cells. In plant cells, though, the same thing happens. Let's say you had 2% outside, 10% inside. So the cell is considered to be in a hypotonic environment. So what's happening is water is moving from the area of low solute concentration to an area of high solute concentration. It's going to swell, but it actually creates just a turgor pressure, okay, which is the pressure pushing along the cell wall of the plant cell. The cell wall prevents it from bursting though. So plant cells will not burst, but animal cells will burst. Okay, this is called turgor pressure in plants, and this is when it eventually bursts. It's called a lysed cell. Now, if you put them into isotonic solutions, go ahead and get them. Isotonic solution right here, then that would be like as if we have 2% sucrose solution right inside the cell and then 2% sucrose solution outside of the cell. Okay, what's happening here is you're putting a cell in the same solute concentration that it's in. Okay, the cell going into a 2%, it has 2% inside. So what's really going to happen is it's already reached equilibrium, so water is going to go in and out just like normal because the molecules are constantly moving, but they're not going to go one way or the other because it doesn't have to even anything out. The last type of tonicity, that you're going to put a cell in is going to be this actual hypertonic solution. So hypertonic means that the solution or the solute concentration that you're putting the cell in is higher than the one in the cell. So that'd be like if we have 10% sucrose solution outside the cell and let's say 1% inside the cell. So water, once again, wants to go from an area of low solute concentration to high solute concentration to try to even it out. So water is going to be actually removed from the cell, and so it causes the cell to shrivel up. And this is once again in animal cells. In plant cells, this is called plasmolysis. When you see a plant kind of withering down and withering away, that's plasmolysis. The cells are kind of corroding and basically caving in, kind of like how our cells are shriveling. Plant cells shrivel up too, but this is called plasmolysis in plants. So you do want to know what happens to the cells in each of these different situations. Okay, try to use flashcards or try to figure it out and be able to figure it out on your own. But ultimately, you should know what happens to an animal cell in a hypertonic solution, what happens to an animal cell in a hypertonic solution, and what happens to an animal or plant cell in an isotonic solution. We talked about certain molecules and so forth that can be transferred across the plasma membrane through diffusion and also osmosis. Some type of molecules or ions cannot cross a lipid bilayer without the help of transport proteins. Okay, transport proteins allow for a specific substance to cross the actual membrane itself. There's two different kinds of actual transport proteins. It can either be a passive transport or it can be an active transport. So passive transport requires no energy to be input. It's completely driven because of the concentration gradient. The actual solute is going to go from an area of high solute concentration to an area of low concentration. Okay, an example of passive transport would be osmosis, diffusion, and facilitated diffusion as well. Facilitated diffusion just means that it gets a little extra help by using a channel or a carrier protein. So in this case, right here, we actually have a channel here. Channel is going to be where it comes straight through, doesn't have to have any kind of assistance. Or a carrier protein is going to be one where a actual transporter protein binds to the actual molecule coming through, and then it changes the shape of the actual molecule until it actually is able to expel it on the inside of the cell. So right here we have a molecule coming in, it's binding right here, changing the shape of the actual transport protein, and eventually allowing that molecule to cross into the inside of the cell. Active transport, unlike passive transport, does require input of energy. It actually moves a solute against its concentration gradient, not with it. So normally, we go from a high concentration to a low concentration because it's trying to even it out in diffusion and osmosis. But in this case, it's going to require energy because we're going against the actual concentration gradient now. So you would think, just by looking at this cell right here, that these calcium ions would want to come across and basically even it out by transferring from a high to a low concentration. Instead, though, you actually see these two calcium ions binding to this transport protein, and then it's going to bind, and then ATP comes along, gets hydrolyzed, and phosphorylates, basically adds that extra phosphate group right here to this transport protein, releasing that energy. Therefore, remember, whenever you break a bond, you release chemical energy. It releases the energy and allows for those calcium to be eventually pushed through against their concentration gradient. Another example of active transport is going to be the actual sodium-potassium pump. Sodium-potassium pump is important to know because it is once again going against the concentration gradient. So here in this little picture you actually see the sodium right here coming across and going through to the exterior part of the cell, and you will actually have potassium coming straight through and come into the interior part or the cytoplasm of the cell. So watch this video and you'll be able to see what's actually happening as far as going against the concentration gradient.
The sodium-potassium pump is an active transport mechanism that is driven by the breakdown of ATP and works through a series of conformational changes in a transmembrane protein. 3 sodium ions bind to the cytoplasmic side of the protein, causing the protein to change its conformation. In its new conformation, the molecule becomes phosphorylated at the expense of a molecule of ATP. The phosphorylation induces a second conformational change that translocates the 3 sodium ions across the membrane. In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid. The new conformation has a high affinity for potassium ions, 2 of which bind to the extracellular side of the protein. The bound phosphate now dissociates, and the protein reverts to its original conformation, exposing the 2 potassium ions to the cytoplasm on the inside of the cell. This conformation has a low affinity for potassium ions, so the 2 bound potassium ions dissociate from the protein and diffuse into the interior of the cell.
So just like the video you just saw, you actually saw the sodium-potassium pump in action. Okay? Remember, it's an active form of transport, so it does require some energy to push it through. A co-transporter, which is a sodium-potassium pump, is actually one that allows for the energy of one reaction to also allow another reaction to occur at the same exact time. So the energy that's taken here from this ATP hydrolysis occurring is used to pump not only sodium and potassium ions across the cell membrane simultaneously. When this ATP is hydrolyzed, basically it's saying it's removing this phosphate group, okay, and it becomes ADP plus another phosphate. This phosphate group binds to this transport protein, and that's called phosphorylation. When the phosphate group binds to another ion or molecule, phosphorylation. So, we have talked already about how some ions and molecules can come across through the plasma membrane itself, but now let's go ahead and talk about how else it can do it for other molecules that are way too big to do it as diffusion, osmosis, or facilitated diffusion. Okay. So you can actually also allow things in and out of your cell with exocytosis or endocytosis. Exo, remember EX means exit. So it's the exiting of a vesicle with those contents inside of it. Okay, so what's happening is, let's say this is the inside of the cell, it actually has a vesicle that's actually surrounded with a bunch of, let's say, cell debris or anything that needs to be removed from the cell. That will actually eventually fuse to your plasma membrane and therefore eventually allow that release of the contents to the extracellular area of the cell. Sorry about that. So that's exiting of contents in the cell. And let's talk about how we would bring things into the cell itself. So into the cell we have endocytosis, okay? It's when a vesicle is formed with the plasma membrane and eventually will enclose the materials that need to come in and bring them into the cell. So right here, being an example, it's actually the plasma membrane is enclosing around those contents that are coming in and eventually bring it into the cell itself. Okay, there are three different forms of endocytosis. There's phagocytosis, which is where there's actually a much larger particle that needs to come into the cell. This is engulfed with a pseudopod, but it's basically a phagocytosis is a much larger engulfment of debris or any kind of particles that are coming into the cell. Penocytosis, which means a much smaller amount, is the same exact thing where a vesicle is coming and a particle is coming and it forms the vesicle to be engulfed into the cell, but it actually allows for a much tinier particle to come into the cell with a drop of extracellular fluid along with it. So this is penocytosis, this is phagocytosis, and the last form of endocytosis is considered receptor-mediated endocytosis. As all receptors do, they're right there waiting for some sort of culprit or substrate to come and bind to the receptor itself. Once it binds to the receptor, then this vesicle can start being formed to the interior of the cell and eventually form a vesicle that will come and coat all those contents inside of it. Receptor-mediated endocytosis is specific to certain molecules to bind to its surface. That's why it needs that receptor to be very specific and not bind to just any kind of vesicles inside. And what is it actually doing here in the actual membrane itself? Like, why do we have endocytosis, exocytosis occurring in the plasma membrane along with all these other transfer of materials across diffusion, osmosis, facilitated diffusion, all this stuff? What's happening is we're actually able to recycle the plasma membrane contents and continually replace patches of these plasma membrane as needed. New membrane proteins and lipids are made in the endoplasmic reticulum. Then they're eventually transferred and modified in the Golgi apparatus, which is our post office. Remember, it's going to receive, modify, and then send off. So it's going to modify these in the Golgi apparatus. And eventually, finally, they're actually going to form vesicles that fuse with the plasma membrane and then that are actually removed from the cell itself.